In our last video, we spent time learning about the 4 emergent properties of water, and now we're going to spend a little bit of time talking about the consequences of those properties, something water can do, which means that water is the universal solvent, and that means it can dissolve and make all sorts of solutions. So today, we're going to be looking at those solutions.
So, based on water's properties, which we were just talking about, water allows for solutions, suspensions, and reactions. So once dissolved, the thing that got dissolved is called a solute, and the water is referred to as the solvent. So when you make Kool-Aid and you put Kool-Aid into the liquid, into the water is the solvent and the Kool-Aid is the solute. That would be a good thing to be able to identify on a test. So if I were going to make chocolate milk, the milk would be the solvent and the chocolate would be the solute. So the smaller part, or the part that gets dissolved, is considered the solute, the bigger part, the solvent. Okay, so if the particles are too big to dissolve, we call that a suspension. This is a suspension. This matters to biology because a suspension is what your blood is. Your blood is cells suspended in blood plasma. So you have white blood cells and red blood cells and platelets and other things suspended in blood plasma. Blood plasma is the solution. That's where like your sodium and your potassium and iron and other things might be stored. But then the cells that are in there, those are considered a suspension. And so you can actually spin your blood down and you can separate the blood plasma up here from the blood cells, which we call the precipitate. This matters because right now in your environment, blood plasma from patients who have been infected with COVID-19 and have recovered is thought to be helpful, therapeutic to people that are currently fighting COVID-19. Now, this is a controversial topic. Some people think it's working, some people think it's not working, some people think there's not enough testing, some people think, well, who cares, just do it anyway, because if it's gonna help them, if it's not gonna hurt them, maybe it'll help them. So what they're doing is they're taking the person's blood that had COVID-19 and they spin down all the blood cells. The blood cells are what could make you react to the blood. They get rid of all of those and they just give you the supernatant or the blood plasma. They just provide that to the patient. And the idea is that maybe some of the antibodies of the original person, the person that was sick and recovered, some of their antibodies will go into the current patient and they'll help them fight COVID-19 before their symptoms get so bad that they would need a respirator. And so it's kind of like an intervention to try to prevent the worst part of the symptoms from happening. Might be something, if that's interesting to you, that might be something that you would want to look up and see kind of what's happening today in that concept. There are billboards around that say, if you've been infected, maybe you can help. Maybe that's something that needs more research. So this is an example of how we can have a solution like our blood plasma or a suspension like our blood, and we can separate the suspended particles into a precipitate. Also, once we've made a solution like this is where reactions can take place. So we often see reactions taking place in a liquid environment. So when we see a reaction like the breakdown of a protein or the buildup of a protein or the breakdown of a sugar, or the buildup of a sugar, they're often happening dissolved in our blood. Our blood is a very important solution, suspension in our bodies. So the next thing we need to talk about is this very special kind of solution that makes acid or a base. So water can actually split apart. So we can take a water molecule and it can separate into H plus and OH minus. So imagine a separation that makes an H plus and an OH minus ion. Now, I'm not going to ask that you can explain to me how or when water would separate like this. I just want you to understand that from our one water molecule, we can separate into these water molecules or these different ions. from our one water molecule, we can separate into these different ions. When we add something to our water, it can cause this to happen. It can make more OHs. It can make more H+ ions. And so what we say is if we have a solution that has lots of H+, that is called an acid. And you may have heard of some acids before, like stomach acid or battery acid or acetic acid. That's vinegar. Not all acids are particularly dangerous. Lemon juice, citric acid, sour candy uses citric acid and acetic acid and ascorbic acid in order to taste so good. We can also have a high concentration of OH ions. We have lots of these. We call that a base. So in our living environments, we create acids and bases. The pH scale is going to measure whether we're more acidic or more alkaline basic. So the pH scale goes from 1 to 14, with 7 being in the center, and 7 is considered neutral. 1 is acidic, and there's lots of H+ when we are acid. And 14 is alkaline. The reason we don't call it basic is because if I said I have a basic solution, there's a misunderstanding, right? I could mean a simple solution, basic. So we call it an alkaline solution to try to help with the confusion, but then that sort of sometimes creates more confusion. So I'm just letting you know, basic, alkaline. We try not to use basic because that tends to be confusing. We tend to use the word alkaline. The most important thing, though, in a biological system is this idea of a buffer. So a buffer is something that we have that allows us to put in acid or base without really changing the acidity. This is kind of like we were just talking about water and how because of its high specific heat, high heat capacity, you can add lots of heat or take away heat and it kind of keeps the temperature nice and stable. A buffer does the same thing but with hydrogen ions. So you can add things with hydrogen ions or remove them. Basically, a buffer is very, very important for homeostasis, which you remember from chapter 1 is maintaining that internal balance. And so a buffer is one of the ways that we can maintain a homeostasis on our internal pH. The foods you eat, whether you eat acidic food or alkaline food, they're going to try to change your blood pH, and that can cause negative things for your body. An acidic pH to your blood has been indicated in increasing your chances for cancer. So we want buffers in our blood that try to keep our pH really constant because that's going to keep our body healthier. So that's the end of our water and solutions topic, and we will be moving on with more about biological molecules and carbon coming up next.